And I'm happy to introduce Paolo Martini, who is actually our longest distance speaker tonight. I think you came from Miami, right?
Yeah.
So we really appreciate your presence here. It's an honor tonight. And before I hand over to you, I'd like to warn the audience, pay close attention to the content, because I will only allow one question about Omicron afterwards. Okay? Okay, the floor is yours.
Thank you. Thank you. All right, it's pretty clear that I'm not going to talk to you about vaccine, okay? Although I've been three times vaccinated. No, I just wanted to give you an overview of the ability of the use of messenger RNA and the capabilities that we have actually been experiencing with this new technology. And obviously, you know, the vaccine works. Hopefully, we prove right that in fact the booster seems to also have an impact on the Omicron. We'll go there. We'll get there probably very soon. But more importantly, I want to show you something that is very different. It's a different use of messenger RNA in different therapeutic areas, and I want to give you an overview of what we do with messenger RNA in order to make it adaptable for different therapeutic areas and for different diseases. You can imagine that obviously when we talk about infectious disease and vaccine, we really want to engage with the immune system. While instead, if we want to treat rare diseases, we want the messenger RNA to be absolutely immune silent so that we can produce the protein without actually the engagement with the immune system and potentially having an anti-drug antibody against the protein, which is actually the true drug in this case. So what Moderna has been focusing on literally is this, what we call it the software of life. And the reason is that if you think about it, the DNA obviously contains all the information, but the messenger RNA take only the key information into the cytosol to then translate the protein. The mutation that you have obviously in your DNA are carried over by the messenger RNA. And for us, using a messenger RNA that is actually wild type, we can reconstitute a normal protein so that we have the ability to really reconstitute the system, the physiological system, the way it's supposed to be. Obviously, as you know, messenger RNA has been very difficult to work with. I remember my first attempt 25, actually only 30 years ago, to be honest with you, so I am that old. But it was impossible to use because of the nucleases. You have to be very careful. Every time there was a little contamination, the RNA was gone, right? So at the time, it was almost impossible to think of messenger RNA as a potential drug. Right now, I can show you, and obviously it is in the clinic, and I'll show you an example, particularly for rare diseases, where we use messenger RNA in a chronic therapy, and it's working. So obviously messenger RNA, the beauty of messenger RNA is that it not only doesn't go into the nucleus, so it doesn't affect potentially chromosome, anything that is sort of encapsulated into the nucleus, but it gives the ability to really translate any different type of proteins. As long as you are faithful to the sequences and the different elements of the messenger RNA, you can actually have a protein that is naturally localized and correctly localized.
Could you mute your laptop?
It is muted. Yeah.
Okay. Let's try.
Let's try again. So you can translate any different type of protein that can localize in the right organs. The only thing that we do is a codon optimization just to have a better translation of the protein. But the rest stays exactly the same. And so you can see here, these are, for example, many of the different projects that Moderna is working on. There are cytokines, there are angiogenic factors, and also in rare diseases, obviously, we look at intracellular protein, mitochondrial protein. We can do transmembrane protein. It was basically impossible in the past to think of a protein replacement of a transmembrane receptor, right? Now we can do it with a messenger RNA. or with obviously gene therapy, you can do it with the caveat that with gene therapy, you have always the limitation in the dosing, in the potentially the number of protein that you can make, while with the messenger RNA, we don't. We have a vaccine that contains, for example, 9 different messenger RNA in a single lipid nanoparticle, so you can actually create complexes. I'll show you an example for propionic acidemia, where, in fact, we treat patients with both subunits that create a complex that is very stable, and patients have mutation either in one subunit or in the other subunit, but regardless, we can treat both patients because we can combine the 2 different messenger RNA without bothering if they have a mutation in a subunit or in the B subunit, because you replace them both. And so it's easier to actually have a therapy that is a little bit more targeted and, more importantly, common for all the patients with that particular disease. So obviously, we have different requirements for different modalities. At Moderna, we are very ambitious, as you can imagine. We started in 2011. Nobody believed us, but we were very secretive, and people hated us for many years because we didn't want to give out our secret. But we had to work on many different aspects, not only messenger RNA stability, longevity, understanding the degradation, but also the route of administration, how to target different diseases with different lipid nanoparticles that would be suitable for different route of administration. And we were very ambitious because we wanted to treat many different diseases. So we have actually prophylactic vaccine, cancer vaccine. We do intratumoral immune oncology, where we overexpress, for example, immune checkpoint to start to recruit, for example, T cells to the tumors. And obviously, we have a localized regenerative therapy. And that's actually a collaboration with AstraZeneca, and we actually inject directly into the myocardial, into the ischemic tissue of a myocardial infarction patients during surgery, VEGF, that reconstitute basically the vascularization of the ischemic tissues. It's a beautiful data. We are starting now a phase 2B with AstraZeneca, showing that in fact we do some regeneration, and it's incredible. And obviously we have systemic secreted therapies and systemic intracellular therapy. That's more towards genetic disorder. The delivery system is everything, obviously, but the beauty of the messenger RNA, conversely to gene therapy, where obviously you have many different factors in terms of control of the amount of protein that you make, with a messenger RNA, you give X amount of messenger RNA and you get X amount of protein. It's mathematical. So in the end, you can do a true dosing of the messenger RNA with the expectation of a true outcomes, without guessing if you're making X amount of protein or little or a lot. It's always mathematical. 0.1, 0.5, 10, it's allometrically scaled, and it's beautiful in that sense. Now, obviously, the delivery then can be different. So you can do a local delivery, as we do in immune oncology, intratumoral, or for example, for vaccination, we do intramuscular injection, and that's how we activate the immune system. Or we do systemic. When you do systemic, obviously, you have to be very careful because your immune system then is very responsive to that. And that's how we started to understand what we needed to do in order to dampen the immune response to our lipid nanoparticle and eventually to the messenger RNA. Also, one of the things that is pretty clear, this is not gene therapy, and we know the messenger RNA has a certain stability as well as the protein. They have a certain half-life, so we capitalize on that. But after that, what do we need to do? Another dose. When you do, obviously, chronic dosing, you need to be sure that your immune system is not responding to that. Otherwise, your therapy goes away. And that's why I'll show you what type of modification we have done in order to be able to go to chronic dosing. Well, in reality, for obviously vaccination, the immune reactivity is important, right? Starting from the local injection, where you start to recruit cells at the level of the antigen-presenting cell at the level of the injection, but also using lipid nanoparticles that they trigger that type of response. The messenger RNA itself is recognized as a virus as well, right? And so we use a messenger RNA that is non-modified, so the system can recognize as a virus and it goes right after it. For obviously rare diseases, we cannot do that because if RNA is recognized as a virus, it's going to be eliminated. So we modify the messenger RNA, so it's not going to be recognized as a virus, but it's actually recognized as just a normal nucleic acid like a DNA, and is actually easily translated. So I'll show you some example. So as you can imagine, it's a complex platform, and I was telling Peter, the beauty of it is that we have 200 scientists exclusively dedicated in understanding messenger RNA, stability, immunogenicity, lipid nanoparticle, delivery, organ targeting, etc. Then I am a therapeutic area, I represent rare diseases. I only have 50 people working with me, but we have 200 scientists working just to understand messenger RNA and how can messenger RNA can become a drug. Which means that not only we have to learn everything about messenger RNA, structure, primary structure, secondary structure, nucleosides, the type of nucleus reactivity of the nucleosides, etc., but also of the delivery system, type of aminolipids that we want to use, the type of components in the formulation that we want to use, that they are triggering an immune response or abrogating an immune response. And so I'll show you everything that we do in order to get to a final drug that could be easily suitable for infectious disease or for rare genetic disorder where you have a completely different type of response. So I want to give you an overview on how we do our engineering of messenger RNA delivery vehicles, how we make some of our vaccine, how we manufacture messenger RNA that avoid the immune system, and how can we achieve a cell specificity, obviously, with the messenger RNA. Sometimes lipid nanoparticles don't allow you to be very selective on the type of organs, but you can be selective on the translation of your messenger RNA in the different organs, and I'll show you how. So different tissues with different route of administration, obviously. We do intramuscular injection for a vaccine. We use a lipid nanoparticle that are very immune reactive. We do intracardial injection. We know lipid nanoparticle just inside a buffer, and you inject your messenger RNA, and it gets easily taken up by the cardiomyocyte and by the local cells. And in the case of VEGF-A with AstraZeneca, we see a nice revascularization of the ischemic tissues during the myocardial infarction. We do also intratumoral injection. As I mentioned initially, we like to target certain solid tumors that particularly have been sort of silent in terms of recruiting immune cells to the tumor by reactivating the tumor by expressing, for example, immune checkpoint that they're critical for recruitment of immune cells to the tumors. And that seems to actually have an impact on the tumor itself. And obviously for rare diseases, we do IV injection that give us the opportunity really to have a more of a systemic delivery and to target different organs. And more importantly, in this case, the lipid nanoparticle has to be immune silent. So let's start because obviously I'm pretty sure that many of you are interested. I'll give you a little overview of how we made the vaccine, but that is the prelude on how we actually then transform the messenger RNA to be anti-vaccine, so to speak. So how our messenger RNA works? Simple. We have a messenger RNA encapsulated in lipid nanoparticle. Generally, what we do, we look at the sequence. We know what the spike protein is. And if we have to encapsulate many different messenger RNA because the spike protein, in fact, is a complex, we can do that. We have a couple of vaccines that either contain 7 or 9 different messenger RNA that can create a nice complex. The beauty of the messenger RNA technology compared to the old technology of using DNA or peptide, it gives you the opportunity to create an incredible repertoire of antibodies, because at this point, if you create a complex, you know that your immune system is going to react to the whole part of the protein, not only to the little peptide or a piece of the DNA that you encoded in your vector. In this case, you can really create the full complex, and that's why the coronavirus vaccine has been very successful, particularly in covering many different mutants, and we'll see about the Omicron, but because in reality, you have the ability to create a full spike protein with the ability to react with different type of antibodies in many different regions of the spike protein rather than just a small region. So once the messenger RNA, let's see, is delivered, obviously, in this case, I have the example of the SARS-CoV-2, the spike protein is being made and is presented to different type of cells, is presented to B cells, and is also presented to different T cells for memory, etc. Once the plasma cells are presented to it, they start to make antibodies, and then we have, in fact, fragment also that can be expressed at the level of the major histocompatibility complex that also allow the recruitment of T cells. I think it's going by itself, and I don't know why. I don't know how to stop it, but it's okay. So in reality, we do actually have the ability to trigger the immune response easily to the messenger RNA by engaging with different immune cells that not only create the antibodies, but more importantly, create the memory. And I remember at the beginning when we started the clinical trial, the big question was because people were tested with antibodies that they finally were able to overcome the coronavirus by themselves, but they didn't have a very good memory. And so one of the key questions was, do the vaccine trigger an immune response from the T cells, from the memory cells, so that we can actually have a constant response to it? And indeed it does. And so that's why in the end, the vaccine became successful because not only created the typical humoral response that you would expect, but also create the memory. And that's why the booster, I think, is necessary because this virus is changing very, very much so frequently that we need to be able to, in fact, cover many different areas. And if, as far as I understand, the Omicron has multiple mutations, which may change quite a bit the structure of the protein, and that's where the protein generally, the antibody generally bind to structural protein. And so that's might elude some of the vaccine. We'll see, obviously, the proof is in the pudding. We'll see if the Moderna vaccine will hold to the Omicron as well. Anyway, the lipid nanoparticle obviously are everything, right? For us, is the only way for us to deliver the messenger RNA. And we have different lipid nanoparticles with different, as I mentioned, immune reactivity. And the lipid nanoparticles are very simple. There's nothing special about these lipid nanoparticles. They're made of aminolipids. We have our own library of aminolipids made of sterol, different type of sterol. Cholesterol is part of also of the cell membrane, right? And so what we create basically is a small cell membrane that is easily recognized by many different receptors. We do have phospholipids. And more importantly, we actually have a PEG that they're modified. And the reason why we modify the PEG, and particularly for rare diseases, is that every day we use cream, we use toothpaste that contain PEGs. So our body is very much primed to IgG against PEG, right? So you can imagine if you give a lipid nanoparticle into the system, all your anti-PEG, IgG anti-PEG are going to obviously attack the lipid nanoparticle by hindering the lipid nanoparticle to deliver to the cells. So what we have done, we modified the PEG and we made it hydrolyzable. So the moment that the lipid nanoparticle enter the system, the blood is aqueous, and being hydrolyzable, they shed directly from the lipid nanoparticle. And so even if you have a circulating IgG, the IgG will bind a free PEG and not a lipid nanoparticle, leaving the lipid nanoparticle free to go to the organ or the tissues that needs to go to. And that's quite easy escamotage, if you will, of using the chemistry of our body to actually have a functional lipid nanoparticle. We do the same for the aminolipids. In the case, for example, of rare diseases, we use aminolipids that they are ionizable and hydrolyzable, meaning that the moment that the lipids are taken up by the receptor, and I'll show you how, because they're ionizable, they get introduced into an endosome. The endosome starts to acidify. acidification with highly charged, obviously, aminolipids has the ability to basically break the system because you have a surge of charges, and it breaks the lipids by releasing, obviously, the messenger RNA into the cytosol, where we want the messenger RNA to be, so that it can be translated by the ribosome, right? And so by using ionizable lipids, we use the system, once again, the pH of the system to get our messenger release. We make them also hydrolyzable, meaning that the moment that they are releasing to the cytoplasm is aqueous. The lipids are hydrolyzable, so they get degraded very fast. So we don't have any accumulation of lipid into the cells because they are degraded by the aqueous system, but we have release of messenger RNA, which is actually our drug. And so in case of rare diseases like glycogen storage type 1 disorder, where the liver of these patients is extremely steatotic because it's engulfed with triglyceride, can you imagine treating a disease, a liver disease with lipid nanoparticles by giving extra lipid to a liver that is steatotic? I can't. But we have done it, and the things that happens is that because of the type of protein that we encode, they actually, not only the lipid nanoparticle disappear, but the triglyceride get deployed as a result of the protein that is given as a replacement. So in reality, we see the mice, for example, from a steatotic liver, 24 hours later having a normal liver. Red, nicely, full of blood, full of activity, and with no issues, and the lipid nanoparticles are gone. So this to tell you that everything that we do for this is not chemically modified, is naturally modified to use the system to assist us in what we want the drug to be. We want an immune reactive, we use a normal messenger RNA, we use aminolipids that are immune reactive. We want a non-immune reactive, we choose particular aminolipid that seems to be nicely recognized by our body. But then we choose also those ones that they are highly positively charged so they can release easily the messenger RNA into the cytoplasm and then get degraded by the aqueous phase. So it's a clever way without chemical modification. So this is what the lipids nanoparticle actually look like, and this is the amount of molecules that we have per lipid nanoparticle. So you can see that generally, depending on the size of the messenger RNA, we can encapsulate between 2 to 6 messenger RNA that are a medium size and between 3,000 base pair. If you go lower with base pair, obviously you can encapsulate many more messenger RNA. But then you have about 6,000 phospholipid, 30,000 ionizable lipids, 20,000 sterile molecules, and obviously PEG. And so this is how a lipid nanoparticle looks like, and it is very much like a cell membrane with encapsulated messenger RNA. How does lipid nanoparticle works? How do they get delivered to the cells? In a very simple way. The moment that the lipid is shed from the lipid nanoparticle, this lipid has the ability to bind to ApoE in circulation. So by binding to ApoE, they get easily recognized by the LDL receptor. And that's why we can actually have lipid nanoparticle internalized through the LDL receptor into an endosome and then get released into the cytoplasm. So it's a very simple natural mechanism using a very natural receptor that is expressed a majority in hepatocyte, but in many different cells. And that's why in rare diseases, for example, initially we have been focusing on a liver metabolic disorder, mainly because we know that we can target easily hepatocytes and also spleen. Spleen obviously could be a problem, right? Because in the spleen you have all the immune cells, and you can also prime your system with your messenger RNA and the protein. So we need to be very careful in delivering to spleen. And so I'll show you in a little while how we actually abrogate the translation of the messenger RNA in the spleen, once again, in a very simple way. So once the lipid nanoparticle go to the desired cell type, we have many different factors, right? We have, obviously, we need to understand endosomal escape, and I alluded to the fact that obviously using ionizable lipid capitalizes on the acidification on the endosome to release then the messenger RNA into the cytoplasm. Then obviously there's the problem of ribosome engagement. And that is also a very good problem to have because you want, obviously, your messenger RNA to be translated as efficiently or more efficiently, for example, of the endogenous one. And so I'll show you how we do it. And then, obviously, once you resolve all these issues, you can have any type of protein to be made by the messenger RNA. So this is an example, and that's why I brought a video, because it really does depend on the type of aminolipids you use and the chemistry of the aminolipids, right? In this left panel, you can see that there are still a lot of red dots. The red dots represent still messenger RNA encapsulated lipid nanoparticle that hasn't been released intracellularly. If you look at our lipid nanoparticle on this side, you look at GFP, you see that the majority of the red dots are gone, suggesting that not only you deliver efficiently, but actually you release efficiently your messenger RNA into the cells with a beautiful translation of your protein. And if we look at the efficiency, in fact, we have about 25 times improvement in terms of endosomal escape compared to the lipid nanoparticles that are currently in some of the clinical trial, actually in the market, as a matter of fact, because there are a couple of products that they use those other lipid nanoparticles with a lot of these red dots. So we have about 25 times improvement by just choosing the right aminolipid with the right chemistry. You actually can have a much better efficiency of your system. In terms of ribosome engagement, always looking at the same type of slides, you look at it, we actually get 2.5 times a better engagement with ribosome. And the reason is that we have been looking at the 3D structure of the ribosome and the primary and secondary structure of the messenger RNA. And we do codon optimization that they allow for the best secondary structure that the ribosome likes more than anything else. So we looked in nature at 30,000 genes and which one was the best translated, what were the characteristics of the translation of these messenger RNA, what are the secondary structure there? And we realized that, in fact, the complexity of the secondary structure in some areas, the messenger RNA, actually allow for a much better translation compared to others. And this is the reason why, in fact, certain messenger RNA translate less protein compared to other, depending on the secondary structure and the ribosome entry. So it's about affinity. It's all about affinity. And creating the structure that the ribosome like allows you really for a beautiful entry and a quick translation with high fidelity. Unfortunately, longer a codon sits on, a tRNA sits on a codon, that's not good news. So you want a fast translation, in fact, because it means that it's efficient and well done. If you start to sit a little too long with your codon, with your tRNA on your codon, it's not good news. You generally are creating potentially a protein that is mutated. And so also this allows us, for example, to treat autosomal dominant disorder, right? Because if your endogenous messenger RNA is not as efficiently made like our exogenous, we can certainly use our wild type compared to potentially a dominant negative form that is going to hinder potentially the protein. So by increasing the ability to translate our messenger RNA compared to the endogenous, allow us really to change the level of wild type protein working versus a potential mutant. And therefore, we can treat also autosomal dominant disorder. How do we avoid immune recognition? This is actually very simple. It was not a Moderna invention whatsoever. It has been in literature that basically uridine are recognized by the toll-like receptors, right? So it's a toll-like receptor 7 and 8 that they have the ability to bind uridine. That's why if you put a normal RNA, is generally recognized as a virus, and through the toll-like receptor 7 and 8 dimerization. And also, unfortunately, a normal messenger RNA has the ability to create double-stranded messenger RNA that obviously it doesn't allow for a good translation of proteins. And the reguide system is actually the system that generally recognizes a double-stranded RNA and put it into degradation pathway. So what we have done, we looked at the synthesis, at the natural synthesis of uridine in nature, right? And we found a couple of modifications of the uridine that are part of the cascade of the synthesis of the uridine. So it's once again a natural change that we apply, and we call them pseudo-uridine. They are generally either methyl or methoxyuridine, which are part of the cascade of the synthesis. And by changing a methoxy group or a methyl group, basically that hindered the ability of the uridine to bind to the toll-like receptor 7 and 8, therefore evading the immune system. And that's the moment that you change the uridine, your messenger RNA become immune silent. And so it gets recognized as a normal DNA. And so that's translated. The issues though that the moment that you change the chemistry, obviously you change the secondary structure of the messenger RNA. And by changing the secondary structure, you might hinder the selectivity or the affinity to the ribosome. And that's why codon optimization and the sequences that we put in order to translate the same type of protein, but to have an RNA that is efficiently translated, that is immune silenced, need to really heavily codon optimized. And Peter was asking me, how many sequences do you need now? How efficient is the system of codon optimization in order to get to that best lead candidate? 2 or 3. We started, I remember when I started at Moderna, we were testing 20 sequences in mice all the time, because obviously you want to see that happening in vivo, right? And it took that long and it took that many sequences. Now the algorithm is sophisticated to a point that we take the top 2 or 3 sequences and compare to the wild type, and generally they do actually have a much better resolution than the wild type. So we do actually have now a very good system in place to select the lead candidate to go into then a human. And then this is just an example to show that in fact, the moment that you change a pseudo-uridine, you see the B cell activation, right, is minimized to 0. Also, one of the things that we explored and we found out, and this is, I think, I don't want to say anything bad, but this is the things that makes the messenger RNA technology of Moderna the best, is that even the purification system that we use is very different from others. And it allows, just by purifying the messenger in a certain way, to actually have a much less reactive messenger RNA and lipid nanoparticle to the immune system. Purification, modifications, that allows for an immune silent messenger RNA that allow us then at this point to do chronic dosing. So in summary, I think we have not only messenger RNA that can be used for infectious disease, for cancer, etc., but more importantly, an immune silent messenger RNA that is now evading the immune system by just simply changing the uridine. And with the purification process, we can make really a drug for patients. And I'll show you an example, in fact. So how do we achieve cell specificity? This is actually, once again, very simple natural mechanism. The messenger RNA that we make looks like exactly what a normal messenger RNA would look like. So as a 5' cap, 5' UTR, open reading frame, 3' UTR, poly A tail, right? Simple, exactly what the nature would see. But how do we make a cell specific? So here in the 3' UTR, we have been looking at microRNA abundances in different organs, right? And so there are very specific microRNA in many different organs. So there are specific microRNA for the liver, for the spleen, for the kidney, And so just by doing the analysis of what is the unique microRNA in a single tissues allow us, for example, to encode in the 3' UTR microRNA recognition sites for that particular microRNA where we want to silence the messenger RNA translation in that particular organ. So if, for example, if we don't want to prime the spleen, but we want only to have an effect on the liver, we look at what is the top and best and unique microRNA in the spleen. We encode microRNA recognition sites in the 3' UTR. The moment that the RNA gets released into the cytosol of the particular tissues, you can imagine that the affinity of the microRNA to its own recognition sites versus the ribosome is much faster. MicroRNA recognize the RNA, double strand degradation, no translation in the organs. And that is a very simple way of making a protein specific for a particular organs. Once again, using a natural system without actually perturbing with chemistry anything like that. And so it's true that maybe we don't have the ability to selectively use a lipid nanoparticle, although now I have to be honest, we have been finding lipid nanoparticle, particularly other components of the lipid nanoparticle, that they seems to favor different cell types. And so we are starting to create lipid nanoparticle that they seems to transduce more the bone marrow versus the endothelial cells, so that we can actually become a little bit more specific on the type of targets that we want. And is mainly due to formulations and the combination of the different amino lipid. But if you want your protein to be abrogated in a specific tissue, the microRNA way is a good way to go, because then it's very specific and you can dampen in that particular organs. So I hope that I convince you that we have done quite a bit of work in understanding the messenger RNA. Obviously, I haven't talked about much longevity and stability, because we have a few patents that are ongoing, but we do actually have some modification of the 5' UTRs that they allow us for a much longer lived messenger RNA in the cells, as well as in the modification of the poly-A tails that seems to have an impact on the residency of the messenger RNA inside the cells, which means that longer it stays and more protein per time you have to make, therefore you have a better regimen potentially for patients. So given that, this is our first example there is right now in the clinic, and I'm very proud of it because it's unprecedented that a therapy for propionic acidemia combine two different messenger RNA to create a very stable complex, and has never been attempted before. I think gene therapy has some limitation in putting many more different genes at the same time. We don't. And therefore, this becomes a sort of a novel way to start treating diseases where you have different mutation in different genes and you can replace them at the same time. What is propionic acidemia? It's an organic acidemia, so it's a liver metabolic disorder. And particularly, it's a key disease where, I don't know if you're familiar, but these children, from the very first moment that they're born, they can die because they cannot process any of the amino acid of the proteins, because everything is dependent on this enzyme that has the ability to really transform a propionyl-CoA into methylmalonyl-CoA, and eventually methylmalonyl-CoA becomes succinyl-CoA that enter in the Krebs cycle. It's a mitochondrial disorder. If you cannot process that, obviously you have metabolic decompensation, a metabolic decompensation that leads to death. And more importantly, these children produce so much propionic acid or methylmalonic acid in case of methylmalonic acidemia that has a huge impact on their CNS, to the point that they are unfortunately severely compromised from the very first metabolic decompensation. So there's no therapy for propionic acidemia, because obviously if you want to be effective, you need to be sure that you replace both subunits. And in some of these diseases where you have many subunits, the moment that you start to have a mutation in one and a less expression of one, eventually in time you start to lose the expression of the other because there's not enough material to be made to create the right complex, right? And that's why it's important to have a system that allows us to replace all the mutations, all the gene mutators, so that we can reconstitute the physiology, the physiological effect of the complex. And so the messenger RNA gives you that advantage that you can combine as many as you want, so that you can overcome the issues of different mutation in different genes. And so, as I mentioned, there's no approved therapy. These children go, many of them go into transplantation as soon as 2 or 3 months of life. As long as they reach the 8 kilograms, they can be transplanted, which is terrifying if you think about it. Also because it means that there are these little nuggets and they have this devastating disease and they get a transplant at that age, which give them basically a survival of about 7 years with all the immune suppressant therapy that they had to go through with the liver transplant, right? 7 to 10 years is the lifespan. So in theory, this should be giving us the opportunity to overcome the issues of transplantation, because we are replacing completely the 2 subunits. And therefore, we should be able to eventually avoid transplantation of these children and the reconstitution of the normal physiological activity of the processing of protein and amino acid. I can tell you that, and I'll tell you now before I show you the data because it's exciting. We started dosing the second cohort of patients. The first cohort was dosed successfully. It's safe and well tolerated. There are patients that already receive 8 to 10 doses. There's no anti-drug antibody. They're still responding to the therapy. That suggests that potentially the immune activation that we have, the immune suppression that we have implemented into the system seems to work. But more importantly, it's safe and well tolerated. So now we are looking at the data potentially to see in fact the messenger RNA is efficacious, and hopefully early next year we'll have a sense of the efficacy of the therapy. But the most important thing for us was to test the safety and tolerability of the therapy, and it seems that these patients are fine. And the FDA granted us the opportunity to go to second cohort, which is great because it means that they believe that is having an impact. I cannot tell you more right now because obviously it's an ongoing clinical trial, and the moment that we'll have full data, we'll publish as soon as possible. But what we do, basically, we reconstitute a normal activity of the enzyme. Here, how it's supposed to work, where you have your propionyl-CoA going through the complex and then transforming into methylmalonyl-CoA and then eventually going into the Krebs cycle. The moment that you have a mutation, propionyl-CoA doesn't get obviously transformed into methylmalonyl-CoA, and it gets accumulated and gets transformed in hydroxypropionate. With impact, hydroxypropionate become a propionic acid and has a huge impact on the CNS and on many other organs. It's very toxic. On top of the fact that you start to produce 2-methyl citrate and also many other different metabolites, which not necessarily are good for the body of these children. And so what we have done, initially, we wanted to understand what is the ratio of the 2 different messenger RNA, right? Because you want to be sure that you put enough subunits to support the complex to come together to be stable. We used in vitro studies. We have cells of patients that they either have a mutation in the A subunit or in the B subunits. And when, if you can see these 2 bars, when we do the combination of the 2 messenger RNA, we get the best activity, suggesting in fact that once you have a mutation on one, if you do only one, for example, in the PCCA deficient patient, we put a PCCA, you would have expected to have the same effect, and you don't, so suggesting that the PCCB also is dampened at that point because the other subunit is missing. And that's why in some diseases it's important to be able to replace both and not only one. And same things we have seen in PCCB deficient patients where we put PCCB and we still don't have the same effect as putting the combination. The ratio turned out to be 1-to-1, which was great because then it made us made our life much easier in terms of creating the drug. And more importantly, once again, here show dose responsiveness in both A or B deficient cells, suggesting that regardless, when you combine the two and you do a dose response, you get the same type of amount of protein and activity that you expect from transducing that amount of messenger RNA. Obviously, naturally co-localizing the mitochondria, that's what was important for us to show. And if you look at luciferase here is not co-localized, while the complex is perfectly co-localized in the mitochondria, suggesting that we do make an active protein that translocate naturally into the right organelles. And then we wanted to test the ability of our complex, for example, to lower the level of ammonia, because one of the other metabolites that is accumulated in these patients is ammonia. It's very similar to some of the urea cycle disorder. And sometimes it's difficult to identify the type of ammonia that you have from patients that they have OTC or, for example, or ASS disease versus the organic acidemia. And so there are obviously ammonia scavengers that they used. And as you can see here, we use carglumic acid in orange. You can see that obviously when you use carglumic acid, it does reduce the level of ammonia. If you look at the messenger RNA, it does reduce the level of ammonia as efficiently as carglumic acid. Additionally, it does reduce also all the metabolites, all the substrates. While carglumic acid cannot reduce hydroxypropionate or 2-methylcitrate or anything else, the dual messenger RNA has that ability. And I can show you here, these are the 3 typical metabolites that we generally measure: C3, C2 ratio, plasma 2-methylcitrate, and hydroxypropionate in 5 doses. You can see that there is a consistency in the dose response manner in lowering the level of these metabolites. And this is dose response. So if you use a higher dose, you actually go very low to reduction of this substrate, which is great. And it works even 5 doses. This is in animal studies. This is true, is an hypomorphic model. So has a sum of the wild type. So you don't expect that much of anti-drug antibody happening. But we have now studies that they've gone for 10 doses and we still see efficacy without actually anti-drug antibodies. And more importantly, in propionic acidemia patients, and I'm almost done. One of the things that you have in methylmalonic acidemia patients, for example, you have end-stage kidney disease, which is also a cause for death for these patients. In propionic acidemia, you have a cardiomyopathy. And the cardiomyopathy develop in time, but they have a large heart, and eventually they could die also for death. What we have noticed by treating, we took actually mice and we aged them to a point that they started to develop cardiomyopathy. And what we noticed that by treating the mice with multiple dosing of the messenger RNA, we were actually able to reduce the heart size, which the heart weight, I should say. Which was actually incredible to see because we didn't expect to have that impact. We expected certainly to reduce the substrate, but we didn't understand if it could have an impact even on other organs. And we were able to see actually a reduction in heart weight. This actually was sort of the trigger for us to take the therapy into the clinic, because at that point we knew that not only we have an impact on the substrate, but we also have an impact on the physiology of these patients. And that's why now the messenger RNA therapy is in the clinic. The dosing that we are using in the clinic is 0.3 milligram per kilogram. So it's a very low dose. In every experiment that we have done, we noticed that with messenger RNA, you can dose as low as 0.1 milligram per kilogram and still have a huge impact in terms of efficacy, which is great because, for example, if you think of enzyme replacement therapy, they use 6 milligram per kilogram, right? In some cases, even up to 10. particularly when you start to develop anti-drug antibody, then they up the therapy in order to overcome the level of antibodies. In this case, we don't need to. It's very little amount of proteins. And if you think it is a combination of two different messenger RNA, you can think that in the clinical trial that we are also started in methylmalonic acidemia, we actually started with a 0.2 milligram per kilogram, which is a single messenger RNA, and which give us the latitude really to have quite a bit of dose escalation if we need to. So this to me is incredible, because in my career, I work in many different types of diseases and with many different animal models, and we never achieve the results that we can achieve with the messenger RNA. There are robust effects that gives me the sense that can be easily translated in human. In my previous life, we have seen so many things that, yeah, they had an effect, and then you go into the clinic and you fail, right? You need to have a really good margin of activity in a model in order to be able to translate well into the clinic. So for me, when I see this effect, I have the confidence, I'll keep my finger crossed, that probably this could work in human. So I think, and I want to conclude here, that we have successfully identified lead candidate combination of PCCA and PCCB. I didn't touch base on the duration of effect, because obviously this is in mice. It's 21 days in propionic acidemia mice. This is actually the regimen that we are using right now in the clinic. And so far so good, but I want to see the efficacy data before I can tell you that this is actually happening every 3 weeks as an infusion. But what is important for me was that we were able to drop the level of ammonia compared to carglumic acid, and more importantly, to drop the level of zero biomarker, and has an impact on the heart. All of these retain a therapeutic efficacy with repeated doses, suggesting that once you start to express a protein in the liver, even if you have a knockout, there has been many data, particularly in gene therapy that has been developed by gene therapy. You start to have actually a tolerization from the liver of your foreign protein, right? If you do the right dosing, and somehow your system start to get adapted by the fact that your protein is expressed by your own organs. And we are not accelerating any process here. We are actually using the natural process of translation. We are not accelerating translation. We are not accelerating ways of releasing. We are just using the system as it is. We are not changing the post-translational modification of any of these proteins, right? Everything is naturally made, suggesting that probably the immune system gets used to the fact that even if you are in a knockout situation, by being produced by your own local organ with the right decorations, the right sugars, etc., you might have a better impact in terms of longevity of the expression of the protein, because then the immune system start to recognize it and start to tolerize it. And I want to mention there is a case for Kunoma, which is for Wolman disease, where it was called the Boston Protocol, where they actually inject a very little amount of protein as a protein replacement to actually tolerize this, as the sections are generally knockout, so they would be very reactive to a foreign protein. By just adding a very little in time, you start to adapt to your immune system. With the messenger RNA, it's even easier because it's your own organs producing it. Therefore, the immune system seems to adapt even better to that. So with all of this, I hope that I convinced you that we have, for the moment, a solution and potentially a synergy with gene therapy, right? What I see the potential issues in gene therapy in having AAV, pre-existing AAV antibody, which obviously makes these patients not eligible for clinical trial. They could certainly use this type of approach, right? And vice versa. So in reality, I don't see this as a competitor for gene therapy, but more importantly, I see this as a synergy for these patients that they have the ability to use gene therapy, and then eventually they become refractory to it. They can certainly use the messenger RNA. Or for those patients that cannot access to gene therapy, they could use messenger RNA as a solution for the potential treatment. And with this, obviously, I want to acknowledge everyone at Moderna, because right now, actually, the picture that you see was taken a few years back. It's impossible now to put all the people of Moderna in the same room, because from I started, we were 300 people. Right now, we are more than 2,000. So it's there's been a huge growth, but also a huge demand, obviously, as you can imagine. And with this, if you have any question, I'll be happy to answer. Thank you.
Okay, Paolo, thanks a lot. So I think the moment when your plane came over Europe, your animation mode switched to Italian speed. So that worked fine.
I've been living in the United States for 23 years, but I will never, and I have an American passport, but I will never change my identity as an Italian. I was born and raised in Italy.
So if we keep up the speed, we have time for a couple of questions, I think. Daniel.
Hi, thanks a lot. I have one question, two questions. One as an experimentalist and just another one as a kind of biologist.
Yeah.
How often do you need to give your drug?
Yeah.
I mean, it will differ from mRNA to mRNA, but I didn't get a feeling of what is the range.
So it depends on the half-life of your protein, right? So we do regimen for every type of diseases. For propionic acidemia, now we are at 3 weeks, so we do an infusion. It's actually interesting because if you look at the infusion for protein replacement therapy, Fabrazyme, for example, goes for 4.5 hours, 5 hours with a big volume, right? Our volume is about 20 to 50 ml only, and the infusion time is about an hour and a half. Every 3 weeks for the moment for propionic acidemia. In the case of methylmalonic acidemia, it's been the same because this enzyme has been proven to be quite stable, particularly in human. These are mitochondrial proteins, so they have an important role, and therefore they are pretty stable, right? They're not fast changing. In other cases, and I can tell you, we have been publishing, we have more than 15 publications. In porphyria, for example, we are at 3 to 4 weeks. It really depends on the protein half-life. But also, as I mentioned initially, and I cannot talk about some of the modifications that we make because we are still patenting them. But depending on the residency of the messenger RNA, that's where you get your longevity, right? Because if you can get at least 3 days, generally messenger RNA is 24 hours, 48 hours, then it's gone. But if you get to 72, 96 hours, then you add in terms of protein making per day, and that's where you start to extend your half-life a little bit. Yeah.
No, I had another question, but
yeah.
I won't ask. No, I have an important question as an experimentalist. When I use lipofactamine or Fugine to transfect my cells, would you consider them very bad?
Yes.
And would you mind going into the biotechnology sector, provide us with that technology?
So there is a difference, actually. I didn't make this clear, I'm sorry, but these lipid nanoparticles are designed for human. They're not designed, so these don't work as efficiently in cells as we actually have seen in vivo, right? And so I want to make this distinction because I mean, to be honest with you, the first experiment that we do, if we need to test a messenger RNA, for example, for the codon optimization, translation, efficiency, etc., we use lipofactamine, in fact. Because we know that we get an efficient transduction in cells, even if it's toxic, etc., it's not important. But that's when we elevate to in vivo and we use our lipid nanoparticle because not only they give us a sense of the efficiency, but also the distribution as well. Lipofactamine is not distributing very well. If you take it into an organ, not only is it toxic, but it's scattered, right? With some of these lipid nanoparticles, I probably in the future I should put some of the slides, but I'm always scared because you need to really see the translatability in different species. But we get 98% of the hepatocyte transduced, 98%. If you look at gene therapy, and actually it's in our paper on Nature Medicine for porphyria, because the group that we collaborated with did a clinical trial in porphyria with gene therapy. And they approached us because they wanted to use PBGD as a messenger RNA. And they did actually put pictures of how scattered is AAV spotted versus how homogeneous is lipid nanoparticle delivery. I don't know what's happening in human. I wish I would be able to take a biopsies to actually see what's happening in human. This is only the question mark that we have right now. We don't know. But if you see the distribution of this lipid nanoparticle in a mouse liver, it's phenomenal.
Okay.
But don't use lipofectamine, please, for in vivo. Never.
Thanks
Yeah.
for sharing. I like the comparison between mRNA and your software. If that comparison stands, then it means that your druggable target space should increase dramatically compared to small molecules. Or
Should be, sorry?
Your druggable target space
Yeah.
should increase dramatically.
Yeah.
So my question would be, how do you prioritize and how do you define a druggable target for mRNA therapy?
So first of all, I want to tell you this. We have done, initially we wanted to understand safety and tolerability. And so what we did, we actually encoded 2 messenger RNA for heavy chain and light chain of an antibody with just modifications in the FC portion so that they could actually homogeneously come together. So we can make log scale antibodies, right? So you can imagine the potential of the messenger RNA as a therapy not only to replace the combination, but also to block with an antibody that is naturally produced. There are many issues with some of the old antibody, and I'm thinking of Soliris, I'm thinking of Xolair, where the limitation is weight per amount of antibodies that you need to use, or solubility of the antibodies. If you have your liver producing it, you bypass all these issues, right? And so that's why we look at these other options. Now, how do we prioritize? That has been, particularly in rare diseases, initially we started with liver metabolic disorder because we had lipid nanoparticles that were delivered to hepatocytes. And that's what we were understanding the best. And we wanted to be sure that we would create an efficacious therapy in an area that we knew that we were going to distribute and to have an impact. Now, as we are exploring different therapeutic areas, what's happening also that many companies are approaching us. So that's why we're starting to do partnership. And I was discussing before with Peter, and we discussed before ultra rare, right? If you have a therapy, if you have a gene replacement that works, how unethical would it be not to make it available to an ultra rare population, right? So we have started this collaboration with UPenn, and we call the Institute of Life-Changing Medicine. We are going to treat Krieger in a jar, and Moderna is not going to make a profit. It's just producing the drug because we have a manufacturing facility that can produce 100 different messenger RNA per day in large scale, right? And so that's how we wanted to, what we wanted to do to actually make the platform accessible to many different people and to many different diseases. So for internally, we prioritize for unmet need, for no competition. That's why PA, MMA, sorry. There is some gene therapy in MMA. There are many publications, though, that this vector have induced severe hepatocellular carcinoma in mice, and that's how we decided to do messenger RNA. PA, there's no therapy available, right? GSD1A, which is our next best things, which we're probably starting a clinical trial either this month or in January. 40% of the patients are already positive for AAV antibodies, right? So they are not eligible for a clinical trial. Ultragenyx is doing a trial in gene therapy for them. So this is the prioritization for us right now is based on unmet need for rare diseases. In the future, my hope is that we can create many more solutions also for non-profit, because if we can make it, I don't want to have a patient advocacy group outside my door and telling me why not me, because to me it's devastating, the fact that we could do it but we cannot do it, right? So I want to give the opportunity eventually for everyone to have access, particularly if we can. And then obviously do partnership. We cannot do everything. You guys know the cost of a clinical trial is enormous. It's true that we had a lot of money, we raised a lot of money, but we wanted to be sure that we will create the right therapy. Now we are capitalizing on expanding the ability of targeting different organs. Eventually, my goal will be also to go into the CNS, right? Not only directly, but potentially with a mechanism that allow for translocation into the blood-brain barrier. So eventually we'll get there with time, right? Right now is a little too difficult because we don't understand all the different parts. What we understand is liver, we understand endothelia, we're starting to understand bone marrow, but there's still a lot to learn, right? But partnership to me is another way to give access to patients to the drug. So we are not afraid to do that. As a matter of fact, we're always looking for potential partner because we have, as I said, I have 15 publications in less than 5 years on top journals, right? We have Nature Medicine, Nature Communications, Cell Reports, everything.
Okay, I think we got it. It's up to us. It's up to us. We have to shoot you an email. That's how it's done. First come, first serve.
I want to give it. I want to find partners.
So thank you very much. That was fascinating, and thank you for coming to Bonn. So you already answered basically my question. I was wondering whether you have strategies to target CNS neurons. So is this different also if you do it applied intrathecally, or are the lipid compositions different? So what is your
I'm telling you, we have tried, right? We have tried directly intrathecal and intracranial ventricular, obviously, because right now understanding crossing the blood-brain barrier is a little too complicated. But intracranial ventricular and particularly seems to work. What we have seen is that the formulation that is critical, right? Is not only the composition of the lipids, but the formulation. So depending on the formulation, you have a different triggering of the glia and the astrocytes. And that's where I want to be very careful instead of inducing an inflammatory response in the CNS, that we have the right formulation that allow us then for a delivery into neurons. We have done ocular, we have done everything. It does work. It depends on the formulation. So we are understanding how to abrogate the reactivity to the glia and the astrocytes, even if we had to go directly into the CNS to begin with. Right?
Okay, Daniel.
Great work, thank you. All this neutralizable lipid, ionizable lipid story and optimum at pH 6.5 emerged from the siRNA screening in vivo to the liver. But when you go to muscle, the situation is different. So did you really compare and prove that this ionizable formulation is optimal for muscle, or is it different from what has been found for liver?
That's a very important question. Skeletal muscle is probably one of the most difficult organ to transduce, to be honest with you. We have been interested, obviously, in skeletal muscle diseases like obviously Duchenne and some of the myopathies, etc. I think we can deliver, but we cannot deliver with the same efficiency as other organs. So if you start to have a very low distribution in a skeletal muscle where you need a very homogeneous distribution, then your therapy is not going to be efficient.
Okay.
And we have the experience of Sarepta, right, with oligos, is spotted. When you have to reconstitute muscle fiber to a certain level that they are efficient in working, you cannot be spotty. You need to be very specific and very homogeneous in the way you transduce. These lipid nanoparticles are not.
My question was about immunization, vaccine.
Immunization?
Yeah, vaccine. I mean, do you use the same formulation for vaccine and for liver? And the situation is completely different.
Yeah, so the formulations depends. We do actually use a similar formulation, but what we change some of the components, right? And so the formulation make a change in terms of delivery and distribution. That's for sure. So, but we adapt to the different organs. So delivery it's easy because we have a formulation now that we know that works well and it transduces well. But when you need to go to other organs, the formulation makes a huge difference.
And what about self-replicating mRNA? Are you what do you think of this technology for your application?
I shouldn't comment because it's a conflict of interest, right? And I don't want to be a bad guy here. Let me not comment on that, but we can talk offline.
Okay.
Yeah, thanks. Great presentation. I just have a very brief question. So you said you're employing the LDR receptor in your system. Is there any data on reduced effectiveness in patients with familial hypercholesterolemia for this?
Patient
for
with
the LDL receptor, if you have patients with familial hypercholesterolemia, is there any reduced effectiveness there?
Yeah, we actually have data. We haven't published it yet because hypercholesterolemia always comes up as one of the diseases that we want to work. We tried a different messenger RNA. It does work. It does work very well, in fact. The problem is the competition that is out there, right, of many different factors, including angiopoietins right now, PCSK9, or anything that is used. So my only concern is that what else do we need in this therapy when in reality there are other diseases that they have nothing, right? And there are areas that they are heavily, obviously, targeted by many different companies because it's they could be potential blockbuster.
Yeah, but I meant not only about the treatment for this disease, but only in your system that you use right now, as you're employing the LDR receptor as part of your system. So is there any hindering in reception?
No, as a matter of fact, because I think is the if I understand it correctly, want to understand if the lipid nanoparticle potentially could have an impact on that. No, they don't, as a matter of fact. And I can tell you that we use model of hypercholesterolemia, in fact, to look at lipid accumulations or impact, and we don't see it. But because of the nature of these lipids, right? And so when you have a faster, these lipids are gone by 8 hours. So the benefits versus the risk is always towards the benefit in this case. And by the time you think that you could have an accumulation, they're gone. And in most of the cases, though, I have to be honest, we use a target that they do actually deploy triglyceride, etc. So there's a double benefit right there. And so suddenly you start to see your liver enzyme dropping naturally to a normal level. We haven't done cases where you just add the lipids for the sake of it and see if it has an impact. That I don't know.
I was wondering about the industry standard. Is that Shire in the 80s or is it BioNTech in 22?
Shire is in the 80s. That was Translate Bio, right? And I actually, believe it or not, I was the first one making a messenger RNA from scratch at Shire.
No, I mean, if you show an industry standard, what is it? I mean,
the industry standard is actually not either Shire or BioNTech. I cannot tell you because I don't want to. You can discover it yourself. Those are MC3 nanoparticle. They're called MC3. So you just do your search and you'll see who's using MC3. But the standard is not either Shire or BioNTech. As a matter of fact, this company have been Shire, I think, which then became a Translate Bio. They've been working with MIT and Dan Anderson, and they have obviously developed their own lipid nanoparticle, and BioNTech is the same. They have their own lipid nanoparticle. MC3 I think come from original for Arbutus or Techmira. I don't remember exactly, but certainly it doesn't have the distribution and the impact that we wanted to have, particularly in a liver disorder where you want to have an homogeneous distribution rather than a spotty one. And more importantly, to target hepatocyte rather than Kupffer cells, because one of the things that can happen if you don't use the right lipid nanoparticle, they have the tendency to engulf Kupffer cells rather than actually be transferred to the hepatocytes. So the fact that we were able to establish a mechanism of internalization through the LDL receptor that gave us the opportunity to really understand. And the MC3, they do the same, but once you go into human, I mean, actually, I should say in non-human primate, you start to see the difference in the distribution and the level of translation that you get. And that's why we started with MC3, to be honest with you. We started with, that was our standard. And the moment that we realized that we were not transducing enough hepatocyte in monkeys, we couldn't go into the clinic because it wouldn't have been a good translation.
Short question, short answer.
What do you learn from administration of billions of vaccines to individuals now from this COVID crisis in developing novel products?
So as I said initially, Moderna has been very ambitious, right? The COVID happened by accident. We were not predicting, in spite of the fact that we have now mechanism of prediction of potentially what are the next pandemic, and many companies already are working towards potential new vaccine that could become the next pandemic. So we had a sense that this was coming, but not to that extent. So the year 2020 for us has been hell. I'm telling you honestly. I work in rare diseases. I had to give people also to work in vaccine, and particularly for quality control, we have expertise in mass spectrometry in my group because for rare diseases is important, and we have to give everything. Just so the prioritization has been COVID. That doesn't take away that we have been working 24/7, and I'm not kidding, 24/7. Unfortunately, iPhones are our enemies. Because of the demand that we had, and also the demand on the other diseases. But when the pandemic happened, we had to stop clinical trial, right? Because we couldn't access hospital, and patients couldn't access hospital for safety, particularly severely compromised patients like propionic or methylmalonic acid. And we were ready to go into the clinical trial. We had to stop to prioritize, obviously, the vaccine, and obviously for the safety of the patients. So in the end, it's always a balance, but that's why we are growing, because, and we are many more people now, because we understand the demand. And if it's true that is also working in rare diseases at that point, that's where we implement even further so that we could support many different therapeutic areas. But I haven't slept in 2 years. And so I'm running on empty here. So.
Okay, Dimitrios?
Thank you very much for the fascinating work. I applaud that you go into rare disease, and especially also ultra rare disease. I think this sounds amazing. Yeah. You've already touched on the whole aspect of immunity in this context, and I understand that your platform will not be affected as much by neutralizing antibodies. However, as a T-cell immunologist, yep. I always consider antibodies just the peak of the iceberg. And if you have antibodies, you most certainly have T-cells. What are your strategies to overcome CD8 T-cell immunity?
So right now, we have no strategy, to be fair. And what we have done was initially to what you have seen, but we haven't developed a secondary strategy for a long-term use right now because we are waiting actually to see the results of these clinical trials. So it was already good to see that we didn't have infusion reaction. And it was already good to see that we didn't have any adverse event, or we were able to mitigate the adverse event by reducing, by increasing the time and increasing the volume of infusion, because in the end, that's what's happening. But long term, I cannot tell you right now, because we are actually monitoring this trial. And the good thing is that FDA allow us to do a multiple ascending dose directly, right? And if it works, you are forced to maintain them on therapy, which means that we can monitor the patients in time and see potentially what is the immune system activation in time. Right now, for what I see from the patients that reach even the 10 dose, it's all okay, but who knows later in time, right? So it's almost difficult to do this prediction in animal model because you know that particularly some animal models, they are overreactive. And I remember when I was a child, we were working in protein replacement. You couldn't give more than 2 or 3 doses, and then obviously IgM, IgG, everything was like elevated. You had IgE, everything. It was just a disaster, right? And that's why immune suppression came through, et cetera, et cetera. So far, so good. We don't see it, but who knows if we see it in time. So the contingency plan is eventually we'll go with some of the immune suppressions. But right now, we haven't seen it, so we are monitoring the patients. And I cannot model in any of the animal model we tried. It's just that is not comparable. So for me, the best case is actually to continue monitoring the patients. And in some cases, I think is the only solution, as a matter of fact. But we do have some ideas. So obviously, eventually, if we see something, we'll start with some co-therapy.
Okay, I think the online audience got that there are certain benefits being present here, but let's give them the chance for the final question.
So Sebastian Chirac asks, since many patients may have a loss of function mutations leading to an absent protein expression, the new BA complex induce auto-antibody production after treatment. What is the experience with the neoantigen effects in general and in BA?
So as I mentioned before, I don't know in patients, right? Because we haven't tested a patient that is completely null. And so I don't know. But for whatever we know from the animal models, we start to see a mechanism of tolerization by just using the liver as a depot for the protein. So far as worked well, we have, as I mentioned, we have long-term studies in knockout. We rescue in knockout, lethal knockout that they would die within 48 hours, we can rescue. And we can maintain therapy for a long-term survival, suggesting that obviously you don't produce an anti-drug antibody, you don't prime your immune system for that particular protein. And we think that the reason is due to the fact that is the liver producing. One of the things that to me was very important to see in Pompe disease, and this is a study that Priya Krishnani has done at Duke, using gene therapy they have a huge expression of GAA, which is notoriously very reactive to the immune system. But because it's produced by the liver, it seems like they don't produce anti-drug antibody with gene therapy for GAA. And that for me was a proof that even a very immune reactive protein, and we know the myozyme, is very reactive to the immune system, which gene therapy is not. So suggesting that when you target the liver and you have your protein produced by the liver and they have a huge amount of protein produced, which could easily prime the immune system, they don't see anti-drug antibody. But I think it's because the liver production and the post-translational modification that they're natural.
Okay, thanks again for the talk.